There will be live thing also, no? Questions they will be typing.
Yeah, live, sir. You can start.
Yeah, good evening, everyone. Welcome to this live session for the course Functional Genomics. We have already shared the details for posting your questions, and there are a few questions that are posted on the Google form, which we would be taking it up. I think even in the live session in the YouTube, perhaps we can add the questions that would you can take it up today. Yeah, I hope this the course is useful. You are finding it interesting, and we will have at least one more live session at the end of 4th week. But in between, we'll have the TAs taking, some of the practice sessions they'll be conducting. I'm sure you are finding it useful as well. So I think with that introduction, we can start looking at some of the questions. Is there anything else you want to add, Rashmi?
No, so you have already told to them. And so for today's question, they can write it in the chat window and then we'll be taking it up. And first we'll start with the questions which were already filled using Google form. So those questions will be addressed and then the live questions. And hopefully in the 4th week, as sir mentioned, that there will be another session. So that time probably there will be live interaction. Say, for example, instead of typing, you can ask questions verbally as well. So do join. Yeah.
That's right. Yes. So the second live session we would really make it live, because I was not sure how many of you would have a very stable connection. So I would suggest that those who are interested in a live session, or if you have that kind of a bandwidth for your internet connection, you please write it in the forum that you would like to join. And if you find that would be difficult, you can still write saying that your bandwidth doesn't allow that. So we'll take depending on what the majority says. Okay, because that is after all for the benefit of the students. So let's take up the question that have already been there. One of the student has asked whether this session is available in both language. Right now, since the students are from different parts of the country, right now we are not thinking of sessions that are more than other than English. So this is right now it is in English. In future, if you have a large number of students and large groups within that student, who could be comfortable with any other regional language, we can consider. But the limitation is that I can offer only one regional language other than English. But we'll try to find if any of the TAs can really help with the other languages. That is something that we can consider. But right now, no, it's only going to be in English. There's a question whether this approach can be used for manipulating the genome of pathogenic virus. I mean, this is a technology for editing the genome. Theoretically, this can be used for editing any genome, but the question is that how that's going to be helpful. The normal way people do is that one of the approaches or application that you can consider is that you want to study effect of a given mutation on the tissue function or the function of the organism. So you try to create that mutation in every cell in an organism by editing the germ cells and things like that. That is what people do. Or you can try to create something in certain, for example, a given tissue, and then people study. But virus, yes, you can theoretically, these are all possible, but it would be very difficult for anyone to create mutation in each and every virus particle that is there. So if you are unable to do that kind of a thing, then your application becomes limiting, because there are other viruses that have the normal um, sequence that make them as virulent they are. So that's not going to really make it, unless the mutation that you have created now becomes something like more virulent, then it will outperform others. Otherwise it's not going to really change much. Okay. Um, that The second question is detailed concept of forward and reverse genetics with their applications, requirements, and so on. I think this particular section has been covered quite well in the lectures. So I would urge you to go back and then play these videos relevant to this topic and learn from that. When you talk about forward genetics, is the you are looking at a phenotype like what you see in the humans, and then you know that there is a defect. Like, for example, we often give an example in the course is the sixth finger. Like somebody has got 6 fingers, that is a mutant phenotype. And then you try to find a gene that, if that runs in a family, then you go and do the family analysis and then try to find out which gene and which sequence in that particular gene uh, change results in this phenotype. So that's the traditional forward genetics. So you start with the phenotype and then find the gene sequence that is causing that phenotype, right? Expression. Now the reverse genetics is opposite to that. So you want to study the function of each and every gene in an organism. For example, human, but you cannot work with humans. So you take a mouse as a model to study the human genes, because most of the genes are what we have in human are also there in the mouse, and they do very similar function. So you mutate one particular gene and then see what happens to the organism, right? So this is the reverse genetics. You create mutation and see what happens. In this way, we'll be able to understand. Now, there are certain limitations. The limitations are, if the gene that you are mutating, um, if the gene is very, very essential for embryonic development, you will not get the phenotype at all. Every time you create a mutation in the gene, the embryo will not survive, right? So you're not going to see any phenotype coming out of it. Or if the gene has got redundant function, let's say, meaning redundant in the sense, it doesn't affect the survival of the organism, and you are maintaining the animal, say mouse, in a lab condition which is well maintained, temperature maintained, humidity maintained, food you are giving. But this is the gene that normally functions when there is a, for example, drought, when there is no food, or there is something, there is a stress on the animal, or there's a pressure it has to run. So that you cannot mimic in lab condition, and therefore, even if you delete that gene, then mouse will be all right, right? So that is, you'll not be able to study the function of the gene. So that's, again, it is not useful in the lab condition. So these are some of those limitations that we have discussed in the lectures. I think I would urge that you go back and study or look at that um, lectures. The third or the fourth question is, what are the benefits and steps of DNA RNA sequencing for disease diagnosis? I think this is this DNA RNA sequencing has a tremendous impact on disease in terms of diagnosis, right? If there are familial forms of disease, it's easy to do a gene sequencing and find out whether I am a carrier. If I am planning for a family, then what is the probability that my next generation would be affected? So it is, it has a tremendous impact. And people, you can, having this kind of a disorder, they can also go for what is called as the IVF, in vitro fertilization. And then if there is a 50% chance of my next generation is having a disease, I can go for an IVF method to see that the embryo that is just formed, whether that embryo is carrying the mutation or not. It is, allow them to grow, make a ball of cells and take a few cells out and do the sequencing analysis to see whether that, embryos get the mutation or not. How, people do the amniocentesis, the people do that see the embryo that is even normally naturally present in the mother, whether early stage, whether that carries the mutation. If it's so, then, it is allowed even to go for um, abortion. In India, that is allowed because there is a huge burden on the family in terms of, maintaining the child and so on. So this is, remarkable advantage in terms of disease diagnosis. A lot of diagnostic methods that you see today, including, for example, your SARS, like, COVID cases, when they are talking about whether it's Omicron or any other new, more virulent variant, people sequence the RNA. So it is routinely done. I mean, one of the positive effect of the COVID is that every, district headquarters now has got labs that have either the RT-PCR machine or, sequencing methods that is, available. So that you would have seen even in India how it has changed. So that has a huge impact and in disease diagnosis, right? So I don't see it is an understatement. Okay. So now let's look at some of the live questions. I'll just look at it. It is,
Yeah.
yeah.
So there is a couple of questions have already come, and first person is Vishwaranjan. Sir, I am from Bihar. So I understand that, I mean, because from various part of country and even outside the country, people join. So that's really great, and that's the reason we prefer to provide this course in English, and then different languages transcripts are available. So if one is facing difficulty, one can always go through with that. That should be helpful. And then another query is from Kalpana Rautela. Good afternoon, sir and ma'am. So I have a question whether we will be having RNA expression analysis related session like qPCR experiment design, cDNA synthesis, and related analysis or not. Okay, so sir can add. I'll just, before that, I'll just add that theoretical thing for RNA expression, say for example, microarray or qPCR, that has week 2 lectures have been released already, and there sir has explained the basic principles. So sir, would you like to add something?
Right. So we did cover, like we have not, PCR is covered, and then we have microarray approach has been covered. These both are, quantitative analysis. And we'll also be discussing about this RNA-seq or next generation sequencing applications. That also, again, really gives you the quantitative difference between, two different samples in terms of the expression. So these are all being covered extensively. I think you would like to wait and see the lectures, and then maybe you want to come back if you have any queries, about 2 weeks from now. Yeah.
Yeah. So another question is from Milan something. How is a gene fusion or translocation similar to or dissimilar to a genetic mutation?
Well, I mean, see, genetic mutation is a very broad term. Any change in the genome that affects the gene is called as genetic mutation. So it could be as simple as one base being deleted, or one base is replaced with another base. Therefore, the codon is changed for that amino acid. You have a different amino acid, or it could be the entire gene is deleted, or what you yourself mentioned, translocation, meaning a part of the gene from one chromosome comes and joins with a part of the gene from the other chromosome. So what happens in this case? If this is the promoter, and if it joins, now this gene would express wherever originally this particular promoter containing gene should express. So it is a misexpression. abnormal expression, which is a mutation. And there are cancers that are known, which result because of this kind of a chromosomal translocations. So, obviously this is also a mutation, right? So they are not very different from other mutation. As long as any change in the genome affects the way the gene functions, that should be called as a mutation. That's exactly you said fusion, because this kind of a translocation can also fuse two different genes together, and that could be an abnormal product or abnormal expression, right? That is there. So there's a question from Akanksha. Is it right to say that both forward and reverse genetics help to link unknown genotype with a phenotype? There's a slight difference. What you have written, the unknown genotype with a phenotype. I'm not too sure what you mean by unknown. You mean to say if in the forward genetics, we do not know what is the gene that causes the phenotype. So you use the phenotype as a tool to go and identify the gene. That is the forward genetics, like the way you find genes that is causing, disease in human. But in reverse genetics, really do not know what the function of the gene, and if you delete this gene, what would be the effect on phenotype. So you do a blind, um, approach, in the sense that you take the gene, mutate, and see what happens, right? That is the reverse genetics. So in one, it is unknown gene but known phenotype. In the other, that is reverse genetics, it is a known gene. You have identified the gene characterized. You don't know the phenotype. It is an unknown phenotype. So either it is genotype to phenotype, or it is other way around, phenotype to genotype. So that's the difference. Otherwise, at the end of the day, in both, you are trying to link the genotype with the phenotype. You either start up with the gene and go and see what is the phenotype, or you don't know the gene, you start with phenotype and get to know the gene. So that's what it is. There's the Deepesh. What is the viability of CRISPR-Cas9 in treating human disease? I don't know what you mean by viability, or meaning to say that whether it can be applied. Yes, I mean, okay, let me put it this way, that whether it is, what is gene therapy? The idea is that if you have a mutation, which is causing a disease, like for example, my vision, right? You had to have certain genes expressed in my retinal cells for me to see using my eyes. If the retinal function is compromised due to a defect in a gene, now I am blind. Okay. But if someone can correct that particular gene in the retinal cell, or if I can put a normal copy of the gene in the retinal cells, that makes me a person with vision. That's a great, tremendous benefit, isn't it? That's a treatment. So far, many other classic treatments use drug to affect a pathway or modify a pathway, like cancer and so on. But in gene therapy, the idea is that whether we can either correct the gene defect, or you forget about that mutated or defective gene, you try to put a normal copy of the gene and see whether that can, restore the function. So that's what it is. Now, it all depends on what risks that you carry by doing it. Now, I am blind, and that's the only difficulty that I have. Otherwise, I am perfectly normal. I can live longer, right? Isn't it? But only I am unable to move on my own in a new place because I cannot see. Now, if there is an approach by which I'm changing some sequence in my genome, and if I'm not too sure whether that change that I am making in the genome is restricted to the target gene, or does it alter some other gene or function. If I don't know that, then what happened is that if I'm doing something to restore a function of the gene, but it also activates some genes resulting in, say, cancer, then I have cancer in my retinal layer, then it affects my survival. So every approach people use or develop, they would like to see that that approach does not, or the risk that that approach may give, if it is extremely low, only then, or nil, only then they would consider for any kind of a treatment. So every approach, it has to be, perfected. It should be developed to that extent. When it is ready, then they will do that. But if it is like uh, a disease that I have because of a defect in the mutation, and then I have only 3 months or 6 months left for me to survive, anyway, I'm going to die. But I have an approach which has the potential benefit. So in that condition, people may try. Okay. So it depends on, like, that's what people do with cancer, another thing. Advanced cancer, when you have, even if the drug is not completely, validated, but you can take a risk and do that, because this if it works, great. Otherwise, there's nothing that you are lost. Anyway, the person is terminally ill. You must have seen people using um, the pig heart transplanted to human and so on. You must have read about it, that you know, uh, newspapers and news and so on. So these are all terminally ill patients, right? There are no other options. Now they are trying to see that the method would help. So that is not something that is perfect. So that is the context that I would like to say. Could you suggest some international institutes, universities, research in plant? There are a number of places even in India, but that's I wouldn't take up that question here. In this forum, this is related to more into the topics that we cover. But even in India, there are multiple places, and that's something that you can take off the track. You can write to us. We'll respond to you whatever we know. Yeah. Um, Between DNA and RNA sequencing, which experiment takes less time to conclude and mostly used for such field? Obviously, I don't know, the traditional techniques, if it is RNA, then it used to take more time, because you have to convert the RNA into cDNA, and normally the DNA was sequenced. But, with this all next generation sequencing, another thing, I don't see much difference in terms of the time taken, right? And of course, um, mostly used for research field. Every method that is developed is used for research field. Only some of them are used for in therapy or in application. So research field, every method is being used. Will the genomic DNA change from one organ to the other, another, in case if you are talking about metastasis? Possible, yeah, that's true. What you're saying is, like, we talk about somatic mutations or chromosomal instabilities, that is known in cancer, right? Yeah, so that is something that is possible. What you're saying. Um, um, and they're asking, would you like to know the testing possibilities? There are, people do that, but then there are, what people are trying to do is that not all cancers are same in terms of their genetic signature, in the sense that what are the pathways affected, what are the genes that are activated or inactivated. So if you can um, dissect that particular signature, and we know the signature, what people try to do is that now see which drug combination would work better for this particular signature. So people now trying to look at, can I create the same kind of genetic um, changes in organisms like fly or zebrafish, which is a short lifespan, and you can really model it quickly, and then give the drug and see which combination of the drug in parallel. You can set up several experiments and see which combination has a better control over the disease progression, and then you can, people try. So this is simply what you are saying, that if you know the signatures, whether that can be used for treatment. Yeah. Yeah, biopsy people do because it may not be of immediate benefit to the patient, but then the patient's clinical history and treatment records, along with the biopsy, the DNA sequence or RNA sequence that you have gotten, you can go you can do a correlation study to know whether these kind of signatures that you have in expression profile are in the mutations, which other drugs are ineffective or effective. One can go back and check it out. In long term, they are of help. Yeah.
Yeah, so whatever the questions were asked, all sir has answered. So if you have any other query, please write to us so that it can be taken up. Otherwise, if you don't have any query, then we'll end the session. Yeah, I see that there are no more questions. Okay, so one question is coming something. So let her write.
We currently define cancer by the organ. I think the question is not complete. I don't know whether you are trying to say that right now the definition of the cancer is based on the source, that is the organ or tissue type, and can that be done using genetic I didn't say that gene therapy can help in cancer. I don't know. I think it gets how gene therapy is given. Yeah, it depends. Okay, that's the question, right? So that's what it depends on. I gave the retina as an example, right? People try even to give for muscular dystrophy, right? The muscle. So basically, this you give people use several vectors. One of them is what is called as adeno associated virus. Right? These are all viruses that do not cause any disease that are present in our body, and you pack the DNA containing this gene into the virus, and then you can use that as a vehicle to target the tissue, and then they deliver there, and then you expect them to stay there for some time and express. This is what it is, and that is one way people have been trying to use DNA, viral based therapy. So you can have a look at it. Mm. The question is whether chromosomal disorders can be solved by uh, gene therapy. Yes and no. If the chromosomal disorders led to loss of a gene, the gene is not expressed, then maybe one can try to put a normal copy of the gene and whether it restores the gene. But if the chromosomal translocation led to activation of the gene, which is seen in some other cancers, uh, is going to be very difficult to silence that. These are some of the challenges. Gene therapy is normally for the loss of function. So if you lost the function of one gene, then you can put a normal copy, therefore it can restore the function. I mean, I don't see any difference whether the gene therapy can be used for old people or young people. So as and when we have a good method, we should be able to do it, but there's still a long way to go. Yeah. There are people who are attempting, yeah. So it should not be a problem. If you're using a viral vector, I don't think this is that much dependent on the age group. Yeah, it should work. Recent tools and software used for evolutionary analysis. I think you should wait for some more time, few more lectures you will have. There's a lot of discussion that we would be doing on the softwares as well. But it all depends on the softwares are these are all tools. It depends on what questions that you are trying to answer, right? Depending on that question, depending on the data set you have, the softwares change. It's not that you have software for ready for everything. There are some that are routinely used, like you must have heard about BLAST and other thing, which is routinely used. But especially those that, bioinformatic tools which people use to mine large data set, they develop their own software depending on the question that they ask. These are not like software that is readily available. So, it is like, any other cell biology technique or genetic engineering technique, you have to have tools. So for example, cloning is a tool, but over the years, there are many other methods have developed depending on what you are using. So similar way in bioinformatics tool also, every day people develop new tools to understand how we can get the meaning out of the genome. Right? I think that's it. I think we have pretty much covered all the questions, and we have one more week. And next week we will look at other questions. By then, you would have gone through the rest of the lectures. Some of the questions that you have right now may be answered in these lectures that are yet to come. So do go through them, and in the next live session, we'll have live question and answer session. Okay. I think we can end here if there are no questions. And thanks everyone for joining. Yeah.
Thank you, sir.
Yeah,
Thank you.
Thank you, Rashmi. We will wind up, sir.
Yeah, thanks.
Okay, sir.
See you. Bye-bye.
Yes, sir. Bye, sir.